Hello everyone. Today we are going to start new video series on molecular biology. So we are going to study structure of DNA, RNA, replication, transcription, translation. So there's a long way to go. The first step, we will learn the chemistry of nucleic acids. Nucleic acids are macromolecules. And they are very important because they are part and parcel of our DNA. So DNA and RNA contains or made up of nucleic acids. So why they are important? They carry genetic information. They store and carry genetic information in DNA and RNA. So the basic units, they are made up of nucleic acids. So in fact, we can call this DNA and RNA, they are nothing but they are polymers of nucleotides. So these nucleic acids, they are nucleotides. In DNA and RNA, there are many such nucleotides joined together by certain type of ester bond that we will study in DNA and RNA. So now when you study nucleic acid chemistry, you should know I have introduced here a term called nucleotides. You should know what is nucleotides. Nucleotides are combination of nucleosides and phosphate group. So nucleotides are nothing but phosphorylated nucleosides. Again there is a new term. What is nucleoside? Nucleosides, they are part and parcel of nucleic acid. They are nothing but they are again combination of what is called nitrogenous bases and sugar. So we started with nucleic acids, and nucleotides are nothing but nucleosides and phosphate group. So nucleosides are nothing but combination of nitrogenous bases and sugar. Sugar is always a ribose sugar. We will see when we see the structure. It may be ribose or deoxyribose depending upon the DNA and RNA. Nitrogenous bases are again two types. One is called purines and another one is called pyrimidines. So there are two types of nitrogenous bases. So purines there are two types of purines. One is adenine. So the one letter abbreviation for adenine is A, capital letter A. Another type of purines which are present in the nucleosides are guanine. The one letter abbreviation is G. Similarly, pyrimidines are three types. Cytosine, the one letter abbreviation is C. Uracil, this is a pyrimidine base which is exclusively present in RNA. The one letter abbreviation is U, and the last type is thymine. So don't confuse thiamine. Thiamine is a vitamin, water-soluble vitamin, whereas thymine is nitrogenous base, pyrimidine nitrogenous base. The one letter abbreviation is T. So these are the nitrogenous bases. So nucleosides are made up of nitrogenous bases, purines and pyrimidines, and combination with ribose, a pentose. So this is the basic introduction about nucleic acid. Now we'll see, we know that now nucleotides are made up of nucleosides and they are phosphorylated. We'll see the exact structure. Here we are going to learn the structure of purine bases. We know that there are only two purines, that is adenine and guanine. Both have parent nucleus purine. First, let me draw what is purine base. Then we will see the structure of adenine and guanine. Purine looks like this. So this is a purine nucleus. From this there are two purine derivatives are derived. That is adenine and guanine. You should know the numbering here. Purine numbering starts from this nitrogen atom. So wherever I have written N, it is nitrogen. If I have not written anything, that is means carbon atom. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is a just purine precursor nucleus. From this there are two purine derivatives, that is nitrogenous bases, which could be present in our DNA or RNA. So here I have copied this purine parental nucleus. Let me write the structure of adenine. So adenine, instead of hydrogen in the 6th carbon atom, will be replaced by amino group. So this will become now adenine. So other name here you can see here in the 6th place, 6th carbon atom, there is amino group. So you can also call it as 6-amino-purine. The parental or precursor molecule is purine here. Only amino group is replaced here. Instead of hydrogen, there is replacement of amino group. So we can call it as 6-amino-purine. Similarly, guanine looks like this. In the guanine, there is replacement of amino group in the 2nd carbon atom. So instead of this hydrogen, there will be amino group here. And in the 6th carbon atom, instead of hydrogen, there will be oxygen. So this will become our guanine. Now easily you can name this. So 2-amino 6-oxypurine. So these two are purine nitrogenous bases which are present in nucleosides or nucleotides or nucleic acids or DNA and RNA. Let us study the structure of pyrimidine bases. Again, pyrimidine bases are derived from pyrimidine ring. It looks like this. So this is the parental pyrimidine ring. From this there are 3 pyrimidine bases are derived. So you should know the numbering here. So numbering starts from here. So this nitrogen, this will be number 1. This is number 2, 3, 4, 5 and 6. So let me draw the structure of 3 pyrimidine bases, that is cytosine, uracil and thymine. So here I have copied the pyrimidine ring. The first pyrimidine base, that is cytosine, looks like this. In the 2nd carbon atom, hydrogen is replaced by oxygen, and in the 4th carbon atom, this hydrogen is replaced by amino group. So this is our cytosine. So you can name here 2-oxy 4-amino pyrimidine. So this is our first pyrimidine, cytosine. Similarly, uracil looks like this. Uracil is very easy. Both in 2nd carbon atom and 4th carbon atom, hydrogen is replaced by oxygen. We have oxygen. So this is our uracil. So easily you can derive the chemical name 2,4-dioxypyrimidine. So oxygen in the 2nd carbon atom and in the 4th carbon atom. So in uracil there will not be any double bond here. The last pyrimidine base is thymine. The structure of thymine looks almost similar to uracil. It has got oxygen in both 2nd carbon atom as well as 4th carbon atom. Now this becomes uracil. I will remove this double bond. Now the thymine in the 5th carbon atom, this hydrogen is replaced by methyl group. So this is the only one base which contains methyl group. So this is our thymine. So the chemical name would be 2,4-dioxy 5. In the 5th carbon atom there is a methyl group, 5-methyl-pyrimidine. So this is our thymine. So we studied so far nitrogenous bases, purine base and pyrimidine base. So in the beginning we learned that nucleosides are made up of nitrogenous bases. So we learned this purine and pyrimidine base, and they are also made up of sugar. So now we are going to study the sugar present in the nucleic acids, that is ribose. So the sugar present in the nucleic acids is pentose. Pentose, we know that it is 5-carbon monosaccharide. The specific name is ribose. So if you want to study more about this ribose or carbohydrates, please go and watch my carbohydrate chemistry videos. I'll put it in the description box. So this ribose is beta configuration. So it is beta ribose, beta D ribose. If you want to know what is beta, it is isomerism, and D is also another type of isomerism. Please watch carbohydrate chemistry videos. You will understand what is beta, what is capital letter D. So the sugar present in DNA and RNA or nucleic acid is ribose. So there are two types here. Either it could be ribose, beta D ribose, or beta D deoxyribose, depending upon RNA and DNA respectively. So let me write the structure of ribose here. So ribose is 5-member ring. So this is a 5-member ring. So I am writing the Haworth structure or ring structure. First, let me name the carbon here. So this would be carbon number 1, this will be carbon number 2, 3 and 4, and here it would be 5th carbon atom. So the last carbon atom is always CH2OH. So you know, if the OH is above the plane, we call it as beta sugar. So here the first carbon atom or in the anomeric carbon atom, OH is above the plane. So this is beta. So this is our beta D ribose. This ribose is mainly present in RNA. So as I said earlier, the sugars could be beta D ribose or beta D deoxyribose. So the deoxyribose, the second carbon atom, the hydroxyl group, the OH group, the oxygen is missing here. Only there will be hydrogen. So in the second carbon atom, there will be hydrogen without oxygen. That means this will become our beta D2 deoxyribose, or simply we may call it as deoxyribose. So this type of sugar is attached to nitrogenous base, then it will become nucleosides. So nucleosides means nitrogenous base plus sugars. Now we will study in depth of the structure. We learned now the structure of nitrogenous bases. Now we will see how these nitrogen bases combine with sugar, that is ribose. That will become nucleoside. So nucleosides nothing but base plus ribose. Base plus sugar. I will keep the sugar because this ribose could be deoxyribose or simply ribose. So I will write the structure of combination of one base and the ribose. Here I have taken a base. This is adenine. This is a purine base. Now we will see how this purine base combines with sugar. Sugar is ribose. So this is our beta D ribose. So in the nucleoside there is combination of base and sugar. Here base, here we have purine base adenine. Sugar we have beta D ribose. So usually, so they will be attached to the 9th nitrogen atom of the purine base and the hydroxyl group which is attached to anomeric carbon atom of the ribose. So they will combine here. So this OH and H will be released as water. So I will remove this. So this is now our nucleoside. So we have initially base adenine and the sugar is ribose. So this will become, the name given to this nucleoside is adenosine. So one more thing you need to remember. So here we have named the purine base as well as ribose. The numbering for base in a nucleoside or nucleotide, this numbering should be cardinal number, whereas the numbering of the ribose in nucleoside or nucleotide should be primed number. Otherwise we always land up in confusion. So prime number is nothing but we have to put prime here. So this is first prime, second prime, third prime, fourth prime and the fifth prime. So the number assigned to adenine is cardinal number, simply 1, 2, 3, 4. Whereas the number assigned to carbon atom of ribose is prime number. So adenine base combines with ribose, it will become a nucleoside adenosine. Suppose base is guanine, combines with ribose, so it will form a nucleoside guanosine. These are the purine base. Suppose a pyrimidine base cytosine combines with ribose, it will form a nucleoside cytidine. Uracil if combines with ribose, then it will form a nucleoside pyrimidine. Lastly, if thymine combines with ribose, it will form thymidine. So please note here, purine bases, when they combine with sugar, they will form nucleoside and their naming ends with osine, O-S-I-N-E. Whereas pyrimidine base, when combines with sugar, their name ends with iodine. You can see cytidine, uridine, thymidine. Whereas purine base ends with osine. Suppose this ribose is deoxyribose, then deoxyadenosine, deoxyguanosine, deoxycytidine, deoxyuridine, deoxythymidine. So far we learned nitrogenous bases and sugars, and also we learned nucleosides. Now we will see what is these nucleotides. So nucleotides, in the beginning only I told nucleotides are phosphorylated nucleosides. Now we already learned the structure of nucleosides. Now we should know the structure of nucleotides. So now which component of nucleoside is phosphorylated? That is very important. So this sugar is actually phosphorylated. The ribose is actually phosphorylated. First let me write nucleoside. I will not write the structure of base here. I will write adenine here. So this is our base. So adenine base combines with this ribose, it will form nucleoside. So this is now adenosine. Now nucleotide means phosphorylated nucleoside. We have adenosine here. Now the sugar component of the nucleoside is phosphorylated. So usually, most commonly, the fifth carbon atom, the OH group, CH2OH, so this group is phosphorylated here. So the phosphorylated form of adenosine looks like this. So this is the one phosphate group is attached to fifth carbon atom, that is CH2OH group of nucleoside, specifically fifth carbon atom of the ribose. Now we will not call it as adenosine, we call it as adenosine monophosphate. Now it will become adenosine mono. Here only one phosphate group is attached here, esterified to the fifth carbon atom, monophosphate, or in short we call it as AMP. So this is our nucleotide, the simplest nucleotide. Now another phosphate group can also be attached here. Here you can see another phosphate group is attached here. Now this will become ADP. So till here it is, from here to here it is AMP. Now from here to here it will become ADP. That means adenosine diphosphate. Again one more phosphate group, if it attach, it will become, now this will become ATP. That is adenosine triphosphate. So this is the most useful, this is the energy currency. So this I have explained only for adenine. Similarly, you can explore for guanine, cytidine, thymidine, uridine. So all these bases when combines with sugar we call nucleoside. When they are esterified or phosphorylated, then we call nucleotides. So last thing you need to remember is what are the functions of these nucleic acids. First and foremost, we know that they are part and parcel of DNA and RNA. So in RNA they contains ribose, whereas in DNA they contains deoxyribose. We already studied what is the difference. So we know that nucleotide ATP, which is considered as energy currency. In all metabolic reactions, whether it is protein synthesis or any metabolic reaction, we require energy, that is ATP, which is nothing but a nucleic acid nucleotide or phosphorylated form of adenosine. We have GTP, which is required for protein synthesis. We have UDP, uridine diphosphate. It is required for glycogenesis, bilirubin metabolism, conjugation of bilirubin, and adenine is mainly required for many coenzymes. So these are the coenzymes which may require these nucleic acids. And also, last but not the least, they are the regulator or modulator, especially cyclic AMP and cyclic GMP. So these are some of the few important functions of nucleic acid. So the next video I am going to explain the structure of DNA. If you want any notes about these nucleic acids, please visit my website vijayaMarakala.com. I will put it in the description box, where you can access the simple notes for nucleic acids. If you like my video, please give thumbs up. If you're not subscribed, please subscribe, share with your friends, and click on the bell icon for future notification. Thanks for watching.